Hello, my name is Julie. I'm a veterinary student at the University of Life Sciences, and I'll be presenting gene silencing by RNA interference of voltage-gated sodium channels in salmon lice. So just for some backup information or background information, as we know, the salmon lice has 8 stages in their life cycle, and the gene expression varies from stage to stage. So here I've just put in a photo from LiceBase to illustrate this, where we see the expression of NAV13 from stage to stage. And the mechanism we'll be utilizing in this experiment is RNA interference, which is a cellular mechanism resulting in gene silencing by mRNA degradation. And this process is initiated by exposure to double-stranded RNA. So the genes we're interested in are the voltage-gated sodium channels, as these are important ion channels for signal transferring in the nervous system. And it has been shown to be involved in delta methrin resistance in other species among the arthropod and insect groups. But we want to find out more about how these actually work in the salmon lice. So, and also the salmon louse has 3 separate genes for these channels, making them differ from the other arthropod and insect species. So these are NAV11, NAV12, and NAV13, and is the NAV13 channel we have been focusing on in this experiment. So our method is based on the work by Christiane Eichner and her colleagues from 2014, where they worked on a method for stable gene knockdown by RNA interference in larvae of the salmon louse. But we've had to make some practical adjustments on this. For example, going over to using microtiter plates where we expose the lice instead of their use of Eppendorf tube lids, for example, and using a shaker while incubating instead of a continuous flow through of water to secure water movement to help the lice molt from stage to stage. So this is the procedure. We expose the NAV1 to double-strand RNA on the microtiter plates, as you see here. And as you can see, we have divided the plates into NAV13 and CD4, where CD4 is the control gene that we've used, taken from pigs. So these are sort of the control group. And then we incubate the lice on a shaker, as I mentioned, to secure water movement. And we sample the lice at the stages of NAPLU2 and copepodids. And then we evaluate their gene expression by qPCR. So what we want to find out is, do we successfully achieve gene silencing by this method? And is there a difference between the 3 genes regarding significance for survival? For example, if they are more dependent on NAV13 compared to NAV11, for example. So our approach to this is looking at phenotypical differences between the 2 groups, so the NAV13 groups and the control group exposed to CD4 double-strand RNA. And then, as I mentioned, we evaluate the gene expression by qPCR. So these are our preliminary results. We found no phenotypical differences between the lice exposed to double-stranded RNA for NAV13 and the lice exposed to double-strand RNA for CD4 from pigs. And as you see here on the graphs, we have the graph to your left is a graph based on the delta-delta CQ values, where we compared the expression of NAV13 to the control group. And for the NAPLU2 group, you can see that the p-value is 0.0001, and our p-value threshold was 0.05. So here we found a statistical significance. And we also found a decrease of or reduction in gene expression of the NAV13 channel at 75% compared to our control group. But when it comes to the copepodids, we did not find a statistical significance, as the p-value here is 0.093. But we did find, but we can see from the graph that the gene expression has decreased a little bit, but we have to evaluate this further. And the other graph here are the delta CQ values, where we compared the NAPLU2 group and the copepodid group for both the gene of interest group and the control group. That was maybe a lot of words, but we compared the expression of all the groups to our reference genes. So yeah, we can see the differences between the NAPLU2 and the copepodids. Yes. So our plan further is finalizing the NAV13 experiment. And we also want to do the same with NAV11 and NAV12 to, as I mentioned, see if we can find any other findings. Thank you for your attention.